Hi everyone, in this lecture we will be talking about the cytoskeleton. Let's first introduce the different types of cytoskeletal filaments. We have 3 in total that are found in the animal cell. The first one is the intermediate filament, followed by the microtubules and actin filaments. This figure shows you the relative distribution of the different filaments in the cell and their relative thicknesses. So we can see that the microtubule is the thickest of the filaments and the thinnest is the actin filaments. The intermediate filaments are called intermediate because their thickness falls in between microtubules and actin filaments. We will be discussing these filaments one at a time throughout this lecture. First, we have the intermediate filaments. From this figure, you can see that the relative distribution of these filaments are all over the cytoplasm. These filaments are the toughest and most durable of the cytoskeletal filaments, and as a result, they provide tensile strength, which is an ability to withstand tension without breaking. Because of this, we can find a prominent amount of intermediate filaments in the cytoplasm of cells that are subject to mechanical stress. These include cells like the epithelium of our skin, or cells that need to be very strong, like the neurons. Next, let's take a look at the main locations where we can find intermediate filaments in the cell. In this micrograph, you can see the intermediate filaments colored in blue, and the red lines that you see here are the demarcations between the different cells. The first place where we can find the intermediate filaments are in the cytoplasm of the cell. And here you can see that they form a mesh that surrounds the entire cytoplasm. Next, we can also find them surrounding the nucleus, and you'll notice that there is a high concentration of very dark blue coloration around the nucleus. And this is actually a structure which we call the nuclear lamina. And lastly, we can find them at the cell periphery, or at the sides of the cell. And here, they are gathered at points called desmosomes. These desmosomes are connections between two cells that keep them together. And here you can see an example of a desmosome, this tight bundle of intermediate filaments. And all over this micrograph, you can see the different desmosomes here, 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 and here as well. Next, let's talk about the structure and formation of our intermediate filaments. Our intermediate filaments are composed of fibrous subunits, and as such, we classify this filament as a fibrous filament. Here's an example of the most basic component of our intermediate filament, the alpha helical monomer. And you'll notice that on one end, it has a COOH group, or a carboxylic acid group, and on the other end, it has an NH2 group, or an amino acid group. In order to create the intermediate filaments, 2 of these monomers have to come together in what we call a coiled-coiled dimer configuration. Then, 2 of these dimers come together to form a tetramer. And this tetramer goes anti-parallel with each other, meaning one tetramer is going in this direction and the other tetramer is going in the opposite direction. And one thing that we notice and is unique with intermediate filaments is that the ends of these tetramers are actually uniform, and we can see the amino ends on both sides. Lastly, in order to form the final fragment of our intermediate filaments, 8 tetramers have to combine to form this structure. The interwoven nature of these different tetramers gives the intermediate filament its strength. We can relate this similar to rope that we see in the real world, wherein we see various strands of the rope being interwoven together. Now, these fragments can come together to form our different filaments, which you can see at the bottom of this slide. Next, let's talk about the different classes of intermediate filaments. There are 4 classes in total. 3 of them are cytoplasmic, and 1 of them is nuclear. For the cytoplasmic intermediate filaments, the type of filament is related to the cell type as well. So for example, we have keratin filaments, which can only be found in epithelial cells. These are the cells in our skin, the bases of our hair and our nails, and in our different mucosal linings. Then we also have vimentin and vimentin-related filaments, which we can find in connective tissue cells, muscle cells, and glial cells. Finally, we have our neurofilaments, which can only be found in nerve cells. For all animal cells, Since all animal cells have nuclei, then they all contain our nuclear lamins. Now, our nuclear lamins come to form the nuclear lamina, which is a two-dimensional meshwork especially created by the cell to protect the nucleus. Now, the nucleus has to be very well protected because any damage to the nucleus will usually trigger the cell to commit apoptosis. So we have to protect the DNA at all costs. Now, our intermediate filaments also have some accessory proteins that help support the structure of these cytoskeletal elements. The first one we'll talk about is called plectin, and it's used to connect our different cytoskeletal elements together. In this figure, we can see the intermediate filament in blue and our microtubules in green. And the plectin are the small orange strands that you see here that are connecting the two. Next, we also have our linking proteins, and these proteins' only job is to link our different parts of the cell together. So in this figure, we can see that the chromatin is being held by a linking protein, and this linking protein is touching other linking proteins, and those are connected to our various cytoskeletal filaments. Next, let's talk about the different diseases that are caused by intermediate filament abnormalities. The first one we'll discuss is called epidermolysis bullosa simplex, which is caused by a mutation in the keratin genes. And as a result, there is interference with the formation of keratin filaments. Patients with this kind of disease often find that their skin is highly vulnerable to mechanical injury, so they need to avoid friction at all costs. Otherwise, they will find that their skin starts to peel off at the very slightest touch. In this figure, you can see an example of a normal skin section on the left side, and on the right side, you can see the skin of a patient with epidermolysis bullosa simplex. And this here is the characteristic blisters that you get whenever you touch a hard surface or when there is friction experienced by the skin. Next, we'll talk about a disease called amyotrophic lateral sclerosis, or ALS. This can also be known as Lou Gehrig's disease. Lou Gehrig was a very famous baseball player back in the day, and this disease is named in honor of him. This disease is caused by the accumulation of neurofilaments in the motor neurons. So even though our intermediate filaments can give strength to our cell, if there is too much of them, they can also degrade or interfere with the function of our different cells. In this case, in the neurons, they cause axon degeneration and muscle weakness. This is also the same disease that the scientist Stephen Hawking had. Lastly, we'll talk about the disease called progeria. This disease is caused by defects in the nuclear lamin and in turn, the nuclear lamina. In the top portion of this figure, we can see a comparison between a patient with a normal nuclear lamina and a patient with progeria. And as you can see, the main difference is that in the normal patient, the nuclear lamina is continuous and uninterrupted, but in the patient with progeria, the nuclear lamina has many defects and perturbations. This disease causes affected individuals to age prematurely. And here at the bottom, we can see an individual who has progeria. Scientists don't really know what causes the premature aging, but they hypothesize that it is related to damage in the DNA and inefficient cell division due to the defects of the nucleus. The next type of cytoskeletal filament we will be talking about are called microtubules. Compared to the intermediate filaments, these tubules are relatively long and stiff. This means that they do not have the ability to bend as much as our other cytoskeletal filaments. Their main goal in the cell is that they have an organizing role, and this occurs in all eukaryotic cells. One of their functions is that they create a system of tracts within the cell that are used for the cell component transport. And those cell components can include organelles, vesicles, and very large proteins. A characteristic function of our microtubules is that they have the ability to rapidly assemble and disassemble in the cell. And all microtubules grow out of an organizing center, which is unique among the different cytoskeletal filaments. The first organizing center they grow out of is called the centrosome, which we can find in the non-dividing cell. In this figure, we can see a graphical representation of the centrosome, which we can find near the nucleus of the cell. And here in this fluorescent micrograph, you can see the centrosome, which is the very high concentration of microtubules found here at the center of the cell as well. Aside from this, in the dividing cell, we also have the mitotic spindles, and in cilia, or your flagellum, we have the basal bodies where our microtubules grow out of. Next, let's talk about the structure and formation of our microtubules. Unlike our intermediate filaments, which are fibrous filaments, we can classify the microtubules as being globular in nature, and that is because its different components are composed of globular proteins. The specific protein is called tubulin, and this comes in a form of a dimer, meaning there are two parts to this protein. We have the alpha and the beta tubulin. Together, they have to stack up to form what we call a protofilament. You can think of this filament as a stack of Lego that keeps on going up and up and up. In order to create the full microtubule, 13 of these protofilaments have to come together to form a sort of tube. And inside the tube, we can find that the microtubule is actually hollow. In this scanning electron micrograph, you can see the 13 protofilaments that surround our hollow center. Now, unlike the intermediate filaments that contain uniform ends, our microtubules have what we call polarity. This means that both ends are different for our microtubules. One end ends with the beta tubulin, and this is what we call the plus end, and the other end ends with the alpha tubulin, which we call the minus end. The reason for these names is that when scientists were doing tests in the laboratory on microtubules, they found that the plus end actually grew more rapidly than the minus end. So in vitro, scientists were able to see that our microtubules could grow from both ends. But in the next slides, we will be able to see that in vivo, or in the living cell, this is not the case. Now, our centrosome, which is the major organizing center of the microtubules in the non-dividing cell, can be found close to the nucleus, and it's composed of two centrioles. The centrioles can be found here. Our centrioles are composed of a special type of tubulin called gamma tubulin, and they form our gamma tubulin ring complexes. Now, the centrioles are the starting point of growth for our different microtubules, which we call the nucleation site. And in the previous slide, we mentioned that our microtubules could grow from both ends in vitro. However, in vivo, or in the living cell, our microtubules can only grow from the plus end, and that is because the minus end is attached to our centrosome. Previously, we mentioned that our microtubules are able to assemble and disassemble very rapidly. We call this characteristic dynamic instability, which is the ability of our microtubules to grow and shrink very rapidly in the living cell. In this example, we can see the centrosome, or the nucleation site, here at the center, and we can see that some microtubules are growing out of this nucleation site. Meanwhile, other microtubules are shrinking back into the centrosome. Our microtubules are in constant dynamic instability. This means that microtubules are constantly being created and are shrinking back into the centrosome. However, our microtubules can be stabilized by attaching to a protein or a cell structure in another part of the cell, and we call this as our different capping proteins. So in this figure, we can see that our microtubules are growing out of the centrosome, and they do this randomly until such time they encounter a capping protein. And when this happens, then those microtubules that are attached to the capping protein become our stable microtubules. The other unstable microtubules continue to grow and shrink back until such time that they encounter another capping protein. Now, how does dynamic instability occur? In other words, how do the microtubules grow and shrink very rapidly? The first thing we have to know is that the growth of microtubules depend on GTP hydrolysis. Free tubulin dimers, that means those that are freely floating in the cytoplasm, contain one GTP which is bound to their beta tubulin. And as long as this GTP is bound to the beta tubulin, these GTP-associated dimers bind strongly to one another. This means that as long as they have GTP, they tend to form our different microtubule structures. Now, if there are a lot of free-floating dimers in the cytoplasm, then the microtubule grows very rapidly. And the end of this growing microtubule is composed entirely of GTP tubulin dimers. We call this as the GTP cap, and as long as the GTP cap remains present in the microtubules, the tubule will continue to grow. This time, let's talk about how the microtubules shrink or go back into the centrosome. Our tubulin dimers can intrinsically hydrolyze GTP, and this occurs soon after they attach to the growing microtubule. Now our GDP tubulin, which is the one that you can see here on the end of our shrinking microtubule, associate less tightly with one another. This means that once the GTP has been replaced by GDP, the microtubule starts to become destabilized. And if there are not enough new or free-floating tubulin dimers with GTP, soon the GTP cap becomes lost in the microtubule. And this loss results in the disassembly of the filament. So this is what we can see here. The disassembly starts from the top of the microtubule and slowly makes its way down until it reaches the centrosome, at which point a new microtubule can begin its growth. Now let's talk about motor proteins. Previously, we said that our microtubules act as highways in our cell. And we can think of the motor proteins as the different cars that run along those highways. They are the ones that carry the cargo along these different microtubules. We have two main types of motor proteins, and we call them kinesin and dynein. Now our kinesin only moves one way, which is towards the plus end or away from the centrosome, and the dynein moves towards the minus end or going back to the centrosome. So even though we have two types of motor proteins, their parts are still similar. The first part, which we call the tail, can be found on top of the motor protein, and this is the part that carries the cargo. Now the cargo can be a vesicle, an organelle, or even very large proteins that are not soluble in water. At the same time, they also have heads, which we can find attached to the microtubule, and they use these heads to walk along our microtubule. These heads also have an ATP hydrolyzing or ATPase activity, which will come into play later on when we discuss how our different motor proteins move. In this figure, we can see the different chemical reactions at work as the motor proteins move along our microtubule. First, let's talk about the two different heads. We will call the head here in the front as the front head, and here in the other side we have the back head. Now normally the back head is attached to ATP and the front head is attached to ADP. In order for the back head to move, it has to first hydrolyze the ATP and convert it into ADP. And don't forget that our motor proteins have an intrinsic ATPase activity. So once the ATP has been converted into ADP, this back head can now release from the microtubule, and now it has to rely on the forward head in order to move forward. The forward head, which has the ADP, must then exchange the ADP for ATP. Once the ATP attaches to the forward head, this drives a conformational change in this protein in such a way that the back head moves in front of the front head. And now we go back to our original state, wherein the back head has an ATP, and now the new forward head has an ADP. And then this cycle just keeps on repeating, and this allows for the walking movement of our different motor proteins. Next, let's talk about the different drugs that affect our microtubules. Here we have some examples of drugs that stabilize microtubules in the case of Taxol, and then we have two other drugs called colchicine and vinblastine, which prevent polymerization or prevent the buildup of our microtubules. Since microtubules are essential for organizing the cell, one of their main functions is that they actually organize the cell during cell division. So when we prevent the microtubules from assembling and disassembling, this also prevents cell division. Some of these drugs can actually be used to stop or treat very aggressive cancers. The final type of cytoskeletal element that we'll be talking about is called actin. Now, if we see on this figure, we can see that the actin is concentrated along the cell periphery. And this is because actin is mostly used for the movement of the cell surface. Our actines are the thinnest of our different cytoskeletal elements. They are more flexible and also shorter than microtubules. But in spite of this, the actin can actually be still very strong because we do not find them usually as individual strands. Rather, they are usually found in bundles and networks in our cell. And they also make up a large portion of the cell cortex, which is the scaffold that holds our cell membrane to the cell body. Now in this figure, we can see that the functions of actin can be very diverse, but all of them exhibit the same characteristic, which is cell surface movement. In this figure, we can see a variety of different actin formations. The first one here are actin that can be found in cilia, and then these ones are actin that can be found as contractile bundles, which can be used to allow the cell to shrink. And here we have an example of pseudopods, which can be used for the cell to move. And then lastly, we have an example of a contractile ring, which is an actin structure used to divide one cell into two. This fluorescent micrograph is another picture of our contractile bundles. Next, we'll talk about the structure and formation of our actin. Just like our microtubules, actin are also globular in nature. And here we can see the basic monomer of our actin, which we call actin monomers. Just like the microtubules, actin also has a plus and a minus end, and they can grow from both ends. However, in the plus end, we should note that there is faster growth. Unlike microtubule, however, the actin inside the cell can grow from both ends and not just from one end. Now, actin can also grow and shrink in the cell just like your microtubules. And the way they do this is with the utilization of the compound ATP, or adenosine triphosphate. Free monomers or free actins in the cytoplasm are usually bound with an ATP. And as long as they have this ATP, they have the tendency to form our different actin strands. Once they have bounded to our growing actin strand, these actin monomers also have an intrinsic ability to hydrolyze ATP to ADP. Once the ATP has been replaced by ADP, our actin monomers now have the tendency to disassemble. Now, one of the factors that affect the growth or the shrinkage of our actin is actually the concentration of free actin in the cytoplasm. Now, if there are high concentrations of actin, we can see that there is rapid growth from both ends of the growing strand. That means that on the plus end and on the minus end, new actin monomers are actively being added. But again, take note that in the plus end, the addition of new actin monomers is faster than in the minus end. But in most cases, there is only an intermediate concentration of our actin monomers in the cytoplasm. And when this happens, we have a phenomenon that is called treadmilling that occurs. And here we can see that the addition of new actin monomers and the loss of monomers is actually equal. So a sort of treadmill-like chain occurs in the actin strand. As we mentioned earlier, actin is not usually found as individual filaments, but rather they are found in networks and in bundles. The proteins that arrange them are called actin-binding proteins. So here we have some examples. Some proteins, like your bundling proteins, create bundles, or they group our different actin together. And then we also have proteins that promote cross-linkages of our actin. We also have myosin motor proteins, which promote movement, and some other proteins like side-binding proteins, capping proteins, and even severing proteins in case the cell would like to destroy its already present actin. Another important set of actin-binding proteins are the ones that control our actin behavior. So one set of proteins prevents polymerization or the assembly of your actin, and we call those as thymosin and profilin. And what they do is that they bind to the free actin monomers and prevent polymerization. On the other hand, we also have proteins that promote polymerization, and we call them as formins and actin-related proteins, or ARPs. Finally, let's talk about some substances that affect actin. Just like your microtubules, these substances also prevent the actin from assembling and disassembling very rapidly in the cell. And as a result, This freezes the different cell movements that are given by actin. Some examples we have are phalloidine, which stabilizes fragments or prevents them from disassembling. And then we have cytocalasine and latrunculin, which prevents polymerization or the buildup of new actin fragments. Alright, so that will be all for our lecture today. Here are some references you can check out if you would like to explore more about our different cytoskeletal elements. Thank you very much for listening, and we hope to see you in the next video.